Okay, so good morning, everyone. Can everyone see the slides? Hello. So can everyone see the slides?
Yes, sir.
Okay,
Yes, sir.
okay, fine. So let us start our class today. Before going to our further discussion on the Baker's East, let us look into what we have studied in our last classes. Okay, so if we see in the overall view of the production of the Baker's East, we have seen that the Baker's East is nothing but the Saccharomyces cerevisiae biomass that is being produced. So we start with, we have seen what is the, so we know basically in this case the what is the biosynthetic pathway. So we have seen that, uh, for example, the glucose being the carbon source for the production of the ethanol or the production of the Saccharomyces cerea. So given a particular microorganism, Saccharomyces cerea, the yeast, basically we can use it for the production of both Baker's yeast as well as the production of the ethanol. And the production of both of them actually depends upon what are the conditions we keep it during our fermentation process. Okay, and each of the and both of these fermentation steps or the biosynthesis pathway can be are switchable in nature, right? By tuning the simple change in the concentration of the oxygen, that is presence absence of oxygen, and the concentration of the glucose. So uh, so that what effect that particular effect yeah, that phenomena is actually known as the Crabtree effect, right? So we have gone into the details, and this actually shows the what is the overall, the chemical the biochemical this reaction that is going on, where it is utilizing, for example, the glucose in the presence of the oxygen produces the biomass, and here we have the biomass as the main product, right? And there is a release of the energy, that is the 38 ATP molecules are released in this particular step, which means around 686 kilocalories are released, and this energy that is being ATPs are released are used up for the production of more biomass. Okay, so this is the overall pathways for the, if you are asked to, for example, to write a detailed pathway, so this is the overall pathway. So then we have looked into the different, like what are the fermentation conditions. So once we have done with the biosynthesis pathway, we look into what are the, the carbon and energy source, what should be the ideal pH, what are the nitrogen sources, what are the vitamins and the minerals that need to be added during the fermentation step, so that we can obviously get the improved yield and the productivity of the Baker's yeast. And whatever the intermediates or the products, or whatever the minerals or the vitamins are, the, for example, in this case the cofactors that are being added, that actually depends upon the biosynthetic pathway. So if you look into the detailed pathway, okay, and each and every intermediate step, for example, have some enzymes, all right? So, and this enzymes, activation of those enzymes requires some sort of a cofactors, or some sort of a, the vitamins or the minerals that are there, right? So depending upon that, looking after into the biosynthetic pathway, we can, determine what are the raw materials that are necessary for the production of the Baker's yeast. Okay, and then we have looked into a very detail of one of the studies on, when it is this particular shows the composition. So one of the thing that we have seen in the sugar, in the carbon energy source, is like we are not taking, the pure glucose, because if you go for the pure glucose, generally the whenever you are getting your Baker's yeast, so Baker's yeast we have seen is a low cost, high is a low cost and high volume product. So you need to start with the carbon source material which is cheaply available. Okay, and that's why, the sugarcane and the beet molasses are mostly used carbon source, and they are obtained as a waste product from the sugarcane industries mostly. or from the any sugar industries. Okay, and if you look into the composition, first what we do is we try to analyze and find out the chemical composition of all the components, and we then add the different additives as per the requirement. Okay, to make an suitable media in which the microorganisms, that is a Saccharomyces cerevisiae, can grow and increase its biomass. Okay, so we have looked into other things like the activators that are there for the fermentation, and there are some of the inhibitors that are there, and some of the other carbon source that can be used, like we have sucrose, maltose, those can be used. However, lactose is something that cannot be fermented by this type, by the Saccharomyces cerevisiae, and it cannot be used as a carbon source. So before we moving to the fermentation step, we look into what are the technical challenges. So we need to understand like what are the major challenges that are there in the yeast fermentation process, and how it can be overcome. Okay, by designing or selecting appropriate the bioreactor systems, or for example by carrying out several subculturing steps. So in this case, what we have seen that generally the generation time is what the main factor. That is means the time, generation time is also called known as a doubling time. So what is the doubling time? So it is time period by which the cell concentration increases or doubles is biomass concentration, right? So at the start of the fermentation, it is the 4 hour for the generation time. That means it could double its concentration or biomass quantity within a very short period of 4 hours. But until the end of the fermentation, it reaches to increases to the 8 hour. So we do not get the high yield or high productivity of this particular Vickers yeast. Then there are other problems of the exhaustion of the growth factors, the absence of the exhaustion of the nutrients, especially the sugar and oxygen. And as we have discussed that with increase in the fermentation time, generally the viscosity of the media increases, because there is a production of the yeasts that are there, and with the increase in the viscosity, the the mixing of the sugar components or the nutrient components that are there, okay, that gets limited. And also when the oxygens are spurged, so this is what this whole process is, a aerobic process, right? So it is aerobic fermentation. That means it requires the adequate concentration of the oxygen. But if you have a viscous material, viscous, because of the growth of this yeast, then the oxygen transfer across the bioreactor media components it's challenging. Okay, so that's why these are some of the limitations, and we have to overcome those limitations by changing our the bioreactor process or our bioprocess as overall. So to overcome those, generally yeast fermentation is carried out by several stages of fermentations at a different scale. So therefore, if you look into the overall, so this particular process flow diagram shows the overall fermentation steps, and it combines the upstream, midstream, and the downstream processes. Okay, so we can see that it starts with the initial pre-culture and then development of the seed culture, and those seed culture is used at a first stage of the fermenter to make some quantity. Like for example, we make the first pure culture. So this tells you what is a kilogram of the biomass being produced. Okay, so initially we have microbial stain stored at particular in a particular vial like system. Like we have seen how we can preserve the preserve this type of different microorganisms. So it is either preserved in some sort of a slant, and then it requires a revival of the culture. So in that we carry out the fermentation in a particular flask, and then after we obtain a adequate concentration of the cell biomass, that is being transferred into the first pure culture. Okay, and then into the second seed culture. So that is first pure culture is almost around 2000 kgs, and then at a 20,000 kgs in the seed culture, and then we have another intermediate step called the semi seed, which ranges around the 80,000 40,000 kilograms, and then only the semi seed culture is being used in the industries in the production fermenters. Okay, and which it grows to as a the volume can be, or the weight of the productivity can be around 500 to 250, for example, kgs. So we can see, so by just increasing the number of steps of this type of pre-culturing, actually we can reduce the effect of that the reduction of the growth rate, these generation times. Okay, so that we can maintain this lower doubling time. Okay, and also we make sure that uh, that fermentation means we should not increase the fermentation time in such a way that there is a very drastic increase in the viscosity, which would lead to the reduction of the means the efficiency of mixing, and the mixing of the oxygen gases or the sugar components or the nutrient components that are there. Okay, so this is how we actually carry out the Baker's yeast fermentation process, and once it is being produced, there are several downstream processes that are there to separate out the yeast, and then finally it is being made into the final products. So we looked into the downstream processings. So before that also we have looked the different stages of the fermentations, starting from the flax fermentation, pure culture, intermediate, stock culture, peach, and the trade fermenter. And we have discussed what are the the significance and what is the time frame of the fermentation for each and every step. Okay, so you should be going through these things in detail. Okay, that especially the what are the fermentation stages that are there. Okay, and why it is being carried out at several the several stages of the fermentation in the series, and what is the, for example, the importance and of each and every fermentation step. Okay, so especially the fermentation duration, the period and time for each and every step is actually different. Like the stock fermentation is different, and the peach fermentation than the trade fermentation. Okay, so what is the relevant importance? So those things are of importance for your studies, for your exams. Now, if you look into the, let us look into after the fermentation is done. Let us look into the downstream processings. Okay, so that is what we will start studying today. So here, what is the main product? Our main product is the biomass. And what is the biomass? Biomass is nothing but the that particular yeast, that is the Saccharomyces cerevisiae. Okay, so when the fermentation is being carried out, what is happening? The microorganisms, the yeast are growing. Okay, and they start to form in form of a solid like lump particles. Okay, and so that is your final product. Okay, and whatever the growth media components or other soluble components that are there, and they can be easily be removed and discarded. Okay, so so therefore, the downstream processing is relatively, very easy. Like in the case of the ethanol, what we have seen that uh, this ethanol it was produced, extracellularly by the same yeast, right? And that ethanol that is being produced was dissolved, was soluble in the growth media component. However, we can separate the ethanol by because they have a difference in the boiling point temperature, and ethanol is volatile in nature. Because of that, it can be separated easily, but we require some of the steps. First step was the removal of the cells from the fermentation media, and then we did the several steps of the distillation, filtration, and so on. Okay, but in this case, since your biomass itself is your main product, so the number of downstream processing steps that are required are relatively low. So how these biomass are separated? So generally in the industries we have a continuous centrifuge system. Okay, which is nothing but if you look into the continuous centrifuge, it is nothing but it has a particular cloth like material at the in a particular tank. In a tank it has a cloth like material, and this particular tank from the bottom is connected with a motor, and it is rotated at a the gravitational speed of 4,000 to the 5,000 g. Okay, where g is the gravity. So it is that the speed at which it rotated is 4,000 to 5,000 times of the gravitational pull. Okay, and when it is rotated, what happens? So we have some sort of a membrane, or a very fine cloth which is having in this case generally these clothes have a very fine pores, which are okay, so which doesn't allows the solid material to pass through it easily. So However, it will lead to the decantation of whatever the liquid components that are there. So what happens in this type of continuous centrifuge, when the the biomass at a very high concentration containing all the growth media components are actually inserted in that, and the formation and the yeast or the biomass that is there, it forms a cake on the surface of this cloth, and whatever the water is there, it is squeezed out across these across these membranes that is used. Okay. And further, what happens when, because we are actually providing a centrifugal force, what happens, the cake that forms on that cloth actually compresses and it squeezes out more water or the growth media components that are there. Okay. So this is how, and this is the only sorry, this is the only final step by which we can separate out the biomass. And then this biomass can directly, because this particular biomass is being directly used in the food industries, whenever, for because bakery is used, as the name suggests, is used in the bakery industries for quite long. So this particular product is then dried, or it is then added with adequate concentration of moisture to make different type of products. However, if you see, so we will see what are the products, two type of products generally that are developed using the baker's yeast. But generally it is found that there are that means when we have this type of the fermentation system, after the fermentation, it is found that the density of the cells that are separated is around 1.133 gram per centimeter cube. Okay, so this cube should be in the subscript centimeter cube, and the water content that is present in the cells are almost 62 gram water content per 100 gram of the cells. Okay, so that you can see that the water content is significantly relatively very high. Okay, and that needs to be removed if you would like to have, for example, some product like this, which is like a compressed yeast. Okay, so to get such type of product, you need to squeeze out the water, and then only you can get it something like some material which is mostly like the texture should be like a butter or any other type of materials. Like, it has to be soft, but it should not break. So this is how generally one of the widely used product, the compressed yeast is actually made. And the compressed yeast generally contain, it has a solid content of 27 to 33%, and the rest it has the moisture and the other components that are present. Okay, so it is also added with different oil, like it is added with the soybean oil or the cottonseed oil. Okay, so that it also forms a continuous ribbons of the yeast cake. Okay, so there are so depending upon these characteristics of products that you are going to make, we have to make sure that we have the adequate concentration of the water in that particular yeast. Okay, so generally, means in the centrifugation step, now how many times actually you are passing your final yeast broth after the fermentation? It depends upon what you are going to make. So like during the first pass to the centrifuge, the yeast concentration generally can be tripled. So like, for example, generally, as we have told that uh, after the fermentation, what is the concentration of the yeast cell? The concentration varies from 4 to 6 weight% of the yeast. Okay, so now, for example, at the first stage of the centrifuge, you are you are going to concentrate, and you are going to remove the water. Because this is a continuous centrifuge, so that is you have to yeah, you have to remember that once the cake has formed, it is continuously removed, and it's scraped out, and again the fresh the fresh broth is being added. So so it is told that at each first pass of the centrifuge tube, where the concentration are it can be tripled. So if initially it starts at the 4%, and then at the first pass it will increase to suppose to the 12. Okay, and then it will go to the subsequent higher quantity. So at each pass we are almost tripling. Okay, and generally, the yeast solids are, we require it at a 18 to 20% concentration. So you can find out that maybe in the range of two to three passes of, of the centrifuge would be enough to obtain this concentration of the yeast solids in the uh, in the in the media. So generally, which the components which is having the 18 to 20% yeast solids, they fall under the category called the yeast cream. Okay, so it is like it's like a pasty like material, and have a whitish appearance, and they have the solid content of 18 to 20% yeast solids. So this is what is called the yeast cream. Okay, and it is stored for several days at temperature generally 1 to 4 degrees. Because yeast generally tend to be, if they are not stored under the cold condition, they tend to be rotten and all those things are there. So generally, they are stored at 1 to 4 degrees and for several days without any deterioration in the yeast quality. Okay. Now, so this is what the one of the product called the yeast cream. The second product is called as the compressed yeast. Okay, so compressed yeast, as I have discussed here, so it looks like it's like the block of cake, and further it is means. So in this case, generally, how do we obtain that? So to obtain this, we further actually concentrated in this case not by centrifugation but by the filtration. Okay, so the filtrations are of different types. I will not go into details on that, but the filtration on the based on the method called plate and frame filter press. So what is that mechanism on which it works? So it has a plate and a frame. Okay, and as the name suggests, it has a plate and a frame through which, so frame actually, that frames are of different sizes, which actually allows to pass the media components that are there, and in between the pores we have the yeast being, the solid yeast being actually stuck into this in the frames. Okay, and then there is a press. So what does the press do? So once a cake has formed on the on the top of this frame, there is a press which actually presses this particular cake, and then it squeezes out more water from this particular frame, and it tries to provide adequate pressure. And so those things are calculated beforehand so that it doesn't squeeze out all the water that is present, but a certain concentration of the waters are that is present in this particular cake is particularly removed. Okay, and in this case, so initially we have seen that that is 18 to 20% in the yeast solids. However, in this case, when we have we use this the plate and frame filter paste. Okay, and either other types are like the rotary continuous vacuum filters. So after these stages, what we obtain is a cake which contains 27 to 32% of the solids. Okay, and generally, if you rotary vacuum filter, it is almost similar in the range of 33%. Okay, so this is what forms the basis of the compressed yeast production. Now, so this is how the compressed yeast production is basically taken place by the use of the plate and frame filter press, and we have in this case we increase the solid concentration to 27 to 32%. For the storage of this type of compressed yeast for a longer period of time, the ascorbic acid is used as a preservative. Okay, and there is another product like so this is how we make the yeast cream, and the second product being the compressed yeast. The third product is actually known as the dry yeast, for example. So what is the dry yeast? The dry yeast means in the earlier two cases there were certain percentage, so we were discussing in the terms of the solid content. Okay, means there is some fraction of the solid, some fraction of the moisture or the water that is present in those material. But in the dry yeast, okay, so the complete removal of the water takes place. Okay, so how does this complete removal of the water is done? So there are several processes of, like the freeze drying or the evaporation is done in at a very high temperature, and what happens, whatever, and for a longer duration of time, and whatever the trapped moisture, or so this particular type of compressed yeast, when we obtain at a concentration of 27 to 33%, and from there, so what is a dry yeast? A dry yeast means it is 100% yeast is there. So from we take this type of this yeast cake, especially the compressed yeast, and then we mash it and we dry it at a high temperature until whatever the trapped moisture is present in this type of cake, that is completely removed, dried, and what we obtain is the dried yeast powder. Okay, in this particular case, the it's complete removal of water has been done. Okay, so generally the stability or the shelf life. So shelf life means like we have seen, shelf life means how much shelf life especially is used for the food products which are perishable in nature. We determine the time period or the time frame by which before which it cannot be used for the food applications. Okay, so generally the dry yeast have the higher shelf life and its temperature ranges from the 1 to 2 years. So these are the generally three products of the baker's yeast that are there. Okay, and each have they differ in the presence of the solid content that is there. Okay, and they also differ in terms of the shelf life there. So generally the yeast creams and all those things have some several days of the shelf life. However, if you would like to have some yeast which having the higher shelf life, it is in the case of the dry yeasts, for example. So this is the what overall we have studied. We have studied um, the fermentation biosynthesis pathway for the production of the baker's yeast. Then we look into the how what biosynthesis pathway, and from that we look into what are the the carbon requirement and energy requirement and so on. And then we look into the fermentation steps, like there are series of fermentation processes and steps that are being carried out, or the stages that are there. And then once the product is being produced, like in this case we have the biomass, how we can the recover those biomass through downstream processing techniques. Okay, and then finally, depending upon the type of the products that you would like to make using the baker's yeast, right? So what are the ranges of the solid content that you would like to keep? And for that, there are different types of the unit operations that are there, which can be selected. Okay, so this is overall picture for the production of the baker's yeast. And next, so anyone has any doubts or questions in the part of the baker's yeast? Hmm. So if we have a bit discussion, for example, so can anyone tell how many stages of the fermentations are there in the, for example, if we so we have discussed that. Can anyone tell like why we are carrying out several stages of the fermentation process in the baker's yeast production? Hmm? Animesh? Hmm? Anyone? Chandra Prakash? Rituraj? Hello, am I audible to everyone? Hello, you have to respond, right? Hello, hello.
Yes, sir.
So there is no response at all in the class. Huh? Like this is very bad overall.
Sir, could you please repeat the question?
So the question is like why we are going for the series of the fermentation steps or fermentation stages. Uh, for example, in the baker's yeast production, right? So why we go for the so we discussed like why this is important to have the series of stages. Like we have seen here, we have the flask fermentation, pure culture, stock, peach, trade fermentations. So why is it required?
Sir, I think because at every stage we want to increase the magnitude of biomass concentration, and that's why we are using a number of steps.
Uh, yeah, that is right, but we have to add maybe some more steps, like uh, what is happening generally in the case of yeast fermentation. If we have starting with initial fermentation stages, if we have, for example, so it is having a doubling time of 4 hours, and if we prolong the fermentation, and then it increases to the 8 hours. So to make sure that we are having at the doubling time of in the range of the 4 hours, we increase the number of stages of the fermentations. Okay, so that more biomass is obtained. So if you are having a doubling time of 4 hours, that means if we start with a suppose 10 gram per liter. So after 4 hours, it will be 20 gram per liter. Okay, and another 4 hours it should be 40 gram per liter. Okay, but it is not like this production rate is constant for a longer duration of time. So once you start a fermentation at a particular stage, suppose at after this for like this doubling time is relevant until like 2 hour of the fermentation, and then after in the fourth to fifth hour, the doubling time increases to the 8 hours. Okay, so that means the productivity decrease. So to make sure that it doesn't reaches to this 8 hour doubling time frame, we use another fermentations step. So where in another fermentation, what we are doing, we are having a fresh culture media of the all the carbon sources, energy source, nutrient source are actually present, and again we make sure that the yeast that is there, that has a fermentation time of the doubling time of 4 hours again. Okay, and like this we carry out, so that at the end of the day we obtain the very high the biomass concentrations. Okay, so yeah. Okay, so anyway, we will continue with our next topic today. So, which is on the acetone, butanol, ethanol production. Okay, so this is also a type of solvent fermentation, and in this case, we have the production of the acetone. The acetone here is the organic, the organic solvent, which is also volatile material, and is generally it is used in the industries, especially the chemical industries, for different other applications. And then we have the butanol production, where the butanol is a type of alcohol, and mostly used in as a fuel additives. Okay, so we will see, before we go into the understanding of the biochemical pathways, what are the stages of the production of the acetyl butanol, and what are the raw materials that are used. Okay, and what are the applications. So let us look into the historical perspective, like we have seen in the case of the baker's yeast. So in the baker's yeast we have seen that how and why. So earlier we have seen that because the production of the baker's yeast or the ethanol, for example, for use in the beverage applications, have been done for last several centuries. Okay, and people generally used to combine the two processes and then carried out the fermentation. But we found out that there are like several disadvantages. Once we studied the biochemical pathways or the biosynthetic pathways, we found out if you would like to have industrial production of saccharomyces cerevisiae, that has to be separated. So there should be a separate fermentation system for the ethanol production and separate fermentation system for the baker's yeast production, and that's how actually the industrial scale production of the baker's yeast came up. Okay, so similarly goes with with the acetone butanol ethanol fermentation. So First of all, we look into who developed these methods. It was developed by a chemist, Chaim Weizmann. Okay, and this was developed in the World War II because the utilization or the demand for the acetone acetone during this era actually increased. And why this acetone was in very high demand? So it is because of the manufacturing of the explosive like trinitrotoluene, okay, and so on. So during this time period era, we all know that there has been several advancements in the technological field and the science field. So the requirement of this type of organic solvents actually increases. Okay, and not much the chemical route of production of this type of organic solvents were available during that time. So fermentation was one of the method by which the acetone was was produced, and this acetone was of great demand because of the production of the explosive this TNT. Okay, however, once this World War One and World War Two actually means this particular after this, what happened, the demand for the development of the production of the acetone decreases, but what happened, the production demand for the production of the butanol actually increased. So with the means after the war period, generally less amount of explosives were made and used. Okay, so the demand for the acetone decreased, but we know that in the present scenario we all are in a scarcity of proper fuel derivatives. So butanol was found to be one of the the alcoholic derivative which is having significantly high calorific values. Okay, and it have a potential for the replacement of especially the petrols and the diesel. Okay, so now the main objective. So a lot of studies are there now is being carried out now on the butanol production, and there are several industries, especially they are coming up with the production of the butanol um, for as an fuel additives. And butanol has also been used and tested in several, in several automobiles and the cars. Okay, and it has been found that butanol can be produced can be used efficiently, but there are some challenges like why the butanol has not been still commercialized as a fuel. So the one of the challenge that is there is the cost of the production by the fermentation route. So cost as well as the time period it takes. So that challenges and that issues what are there in the fermentation side, that we will be seeing and discussing it. Okay, in our today's class and on the subsequent classes. So and this is how generally our demand for as the demand for the acetone actually decreased. Now we have a very high demand for the butanol, and this the production of in the acetone and the butanol, actually both of them are produced through a single fermentation process. Okay, and the production of either of the two can actually be switched, like we have seen in the case of the ethanol production and the baker's yeast production. So the production of the acetone, butanol, or the ethanol. So along with so here you can see the name. It is called ABE fermentation because along with the acetone and the butanol production, there is some fraction of production of the ethanols are also there. Okay, so that is all coming out as a byproduct, but we can tune actually, like during the fermentation process, we can tune either we want to go for the production of the acetone, and then we have to provide adequate the environment or the adequate media components which will increase the production of the acetone, or if we are going for a production of the butanol, then we have to provide the adequate environment. So this is we will look into and understand when we look into the biosynthetic pathway. Okay, and along with the production of the acetone, butanol, there was a production of the vitamin B12, which was actually produced as a byproduct. Okay, and we know that this particular byproduct, the vitamin. So there are several vitamins: vitamin B12, vitamin C, vitamin D. All those vitamins can be produced by the fermentation techniques, and most of the vitamins that are you are using for your daily life application in the medicines and so on. So these vitamins are produced by the fermentation route and the fermentation technology. So if time permits, we will look into very briefly we look into what is how the vitamins are actually produced. So let us now look into and understand. So, like, what is the different stages of the AB fermentation? Okay, so if we look into the first of all, let us look into the biosynthesis pathway. So, if you look into the biosynthesis pathway and compare with whatever the pathways you have, for example, you have studied till now, uh, if you compare that, so you will find that the basic fermentation steps remains the same. Okay, so first thing we know that we have seen in the case of the ethanol fermentation as well as the Baker's yeast production, that if we start with the glucose as a substrate, the glucose to the like in the pathway of the glycolysis, uh, in the glycolysis pathway, it will be converted to the pyruvate, and the from this pyruvate we have seen it becomes a branch point, and from the pyruvate we have seen how the ethanol is being produced in the absence of the oxygen, and how the pyruvate is used by the yeast in the TCA cycle, or the target carboxylic acid cycle, to produce the 38 moles of the yeast, and that is being used for the production of the biomass. Same way, when we are having the AB fermentation using the glucose as the carbon source, so we come to a branch point at the pyruvate, and the pyruvate is means there are several branch points actually in this AB fermentation. So this pyruvate is converted to the acetyl CoA, okay, which can be actually used for the production of the acetate, or it can be used for the production of the ethanol. So generally, if you see. The fermentation processes, especially which is related to the acetone butanol fermentation, they are classified into two types. So one is the acid fermentation phase, and another is the solvent fermentation phase. So why it is called the solvent fermentation phase? Because ethanol, acetone, butanol, so they are mostly used as a solvent in several chemical industries. Okay, and that means that is they are required at a very high volume. Okay, and they generally are also having the low cost. Okay, so the production of the solvent can actually be tuned by how we how we add different factors or different components in the fermentation media. And the acid fermentation phase, if you would like to produce, so the acetate means it is going to produce, for example, the acetic acid. Okay, or the butyrate means it is going to produce the butyric acid, for example. Okay, so depending upon if you would like to produce, because acetic acid also have a significant the commercial significance are there. Okay, so there is a demand of the acetic acids. So if you want to produce the acetic acid, then you have to carry out it to the acid fermentation phase. And what are the parameters, governing parameters and the conditions, okay, that is required to switch either of the acid fermentation phase and to the solvent, or vice versa. That depends upon the several factors. So that is what we are going to look and understand those. Okay, so if you look into the stages of the AB fermentation, so generally it can be classified into different phases. So one of the phase is called the acidogenic phase. Okay, another phase is called the solventogenic phase. So I think, as the name suggests, you can pretty understand that what is going to happen in the two phases. Okay, and so generally these are the two stages of the classification. And what happens, so if you see the normal, so if you see the what is, first of all, let us see what are the microorganisms that are that are being used. So the most of the microorganisms which actually are used in the AB fermentation are the Clostridium species. So the Clostridium's particular species, these particular microorganisms are widely used for the AB fermentation, and generally they produced, for example, the hydrogen, carbon dioxide, acetate, or the butyrate during the initial growth phase. So when they are like at the initial stage of the growth phase, and they starts to release this type of the acids, okay, the acid derivatives, okay. And these are actually the products that are formed during the initial growth. And what is going to happen if the increase in the concentration of this type of acetate or the butyric acid is taking place? So generally the pH will actually decrease, okay, and this is going to affect the growth of the microorganisms, and it is going to affect the production of the other solvents that are there, okay. So you can say that this acetic acid and butyric acid are the growth associated products. So we have seen the right the different several classifications, I think in our earlier classes before the mid-sem. So we have seen that there are different different stages of growth at which the products are produced. So if we look into the graph, right, with the fermentation, like it may be the product with the fermentation time, and it may be the biomass concentration. So with the increase in the biomass, the cell concentration, we would find that there is a production of this particular acetic acid or the butyric acid, okay. Until then there is no production of the solvents like the acetone, butanol and the ethanol. So they are generally produced during the stationary growth phase. So we know what is the stationary growth phase. Like once a microorganism have overcome the lag phase and then goes to the exponential phase, and it has utilized all the carbon source that are present initially or added to the fermentation process, then it reaches a stationary growth phase, okay. And during this stationary growth phase, what is it is doing, it is it is maintaining a metabolically active state. That means it is carrying out all the activities like in the metabolic pathway, like it is that is used to that is required to make the the microorganisms alive. And during the stationary phase, it actually produces the solvents, the acetone, butanol, and ethanol. And this particular phase is actually, that's why called as a solvatogenic phase. Okay. So okay, so when these particular solvents are being produced, so what is happening? The acids that are that is a reassimilation of the acids actually takes place, which is being produced in the first phase, right? So that actually leads to an increase in the pH. So earlier, a decrease in the pH because of the production of this acidic component acetate and the butyrate. However, because the reassimilation of the acids are taking place during the second phase, so generally what it will occur, it will lead to the increase in the pH of the culture medium. Okay. Now, if you look into the carbon sources that can be used, okay, so one of the interesting thing is that in this particular case, where the Clostridium species is the the microorganism, we can take the hexose sugars, and the hexose sugars, not only the monosaccharides like the glucose, but the di and the trisaccharides and the polysaccharides can also be used. Okay, so this is because this Clostridium species can produce the enzymes which can break the tri and the polysaccharides into the monosaccharide unit. Okay, and they are generally metabolized with the pathway which is known as embedded Mayer-Rov pathway. And if you look into the, we will look into the overall biochemical pathway, but if you look into briefly on that, so 1 mole of hexose, which is nothing but the glucose or the sugar derivative, is converted to the 2 mole of pyruvate. Okay, and there is a production of 2 moles of the ATP along with the production of the two moles of the NADH. Okay, so this is what the what is the carbon source that is being used. So we are having a hexose sugars, and along means in the terms of the hexose sugars, different di, tri, and the polysaccharides can be used. However, one of the interesting thing is that the pentoses can also be the pentose sugars can also be used for in the fermentation media. So in hexose we have the 6 carbon, right, 6 carbon or the 6 groups, and in this case we have the five, these five particular groups are there. Okay, so generally if you look into those hexose structure, generally if you are going with some of the agricultural biomass as a source of carbon source for the production of the acetone butanol, then so the pre-treatment. So we have seen that if you are going to use the agricultural biomass, if you were to extract the cellulose, so you have to carry out several pre-treatment techniques, and the cellulose can be converted to the monosaccharides by the presence of the cellulase as an enzyme. Okay, but in this case, we don't require those because the microorganisms, the Clostridium species, knows how to produce the enzymes which will actually cleave the di, tri, and the polysaccharides into the monosaccharides. Okay. However, another advantage that it can use the pentose, and pentose are what? So pentose are nothing but that is obtained from the hemicellulose fraction, and they are nothing but it is also called as a xylose. Okay. So given an agricultural biomass, we have already discussed that it has made out of the cellulose, it is made out of the hemicellulose, and it is also made out of the lignin. Okay. So the cellulose part can be used can be used as a carbon source, but generally this hemicellulose part are generally lignin part generally are thrown away. Okay. Now what is happening, because it is able to use the pentose sugars, so the hemicellulose is nothing but it is the backbone of in the backbone it contains the 5 ring xylose structures. Okay. And so pentose can also be used. So both, if you look into the picture, so hemicellulose cellulose can be used, as well as hemicellulose can be used. So what is going to happen, so more conversion of the substrate will take place, and we will have the higher productivity of your of your particular targeted product. Suppose you, in the case only when you are using your agricultural biomass as a carbon source. Okay. So this is the overall. And now if we use the hexose sugar, and obviously the fermentation pathway will change. And what is going to change, we have already seen that if we are going to have some other carbon source, there is some sort of a chemical modifications, and that is being done, and then it actually reaches to the and it reaches, and it becomes a component of the biochemical pathway. So here we can see that well, like if you have the pentose, so generally the pentose is converted to the pentose 5 phosphate. Okay. And then it is disseminated by the means of this trans keto acetylaldose sequence. Okay. So after this particular the chemical reaction step, it actually is used for the production of the 6 phosphate and the glyceraldehyde 3 phosphate, which actually, so at this particular point, in this in this particular point of the fructose 6 phosphate, it starts to produce, means it enters into the biochemical pathway, and then it follows the similar route for production of the acetyl CoA, and from which actually it comes to the branch point. Okay. And now we have to look what is happening is this at this particular branch point. So we will look into this I think in the tomorrow's class. Okay. And there is another change that is going to be there, like your quiz was scheduled, I think, for the in the next week. So we would prepone your quiz on we will prepone it, and we will have a quiz on Thursday. Okay. So so that everyone should keep that in mind, that their quiz has been preponed, and please inform to your friends. Okay. So anyone has any questions or doubts until now? So if not, then let us meet and discuss in the next class. So thank you, everyone.
Thank you, sir.
Thank you, sir.
Thank you, sir.